Okay, tonight we're going to talk about inheritance. So Trent said earlier that inheritance would be those characteristics we get from our parents. Okay, so since we're talking about genetics, heredity, inheritance, no good conversation of inheritance is complete without a discussion of Gregor Mendel. Does anybody know who Gregor Mendel was? Anybody ever heard of Mendel? He was a monk and mathematician who gave us our fundamental basic principles of genetics. So let's go back in time to the time that Mendel actually lived and talk about what some of the beliefs were during that time. Okay? So in the 1850s, when Mendel was doing his work, the most accepted explanation of why offspring were the way they were was something called the blending theory of inheritance. Okay? Keep in mind, this is 1850. We don't know a lot. Okay? We obviously do not know anything about chromosomes or genes or DNA. We barely know about cells. Okay? So humans really didn't understand how offspring received the characteristics that they did. They had figured out that to have an offspring, it takes a man and a woman. That didn't take long to figure out, okay? But they really didn't know what the contributions of the female were versus the contributions of the male, okay? So they believed this strange theory called the blending theory of inheritance. So to explain the characteristics of the offspring, they thought that the qualities of the parents blended in the offspring, okay? The qualities of the parents blended in the offspring. So if you had a tall parent that mated with a short parent, you would blend that, you would get an offspring of medium height, okay? Or if you had a red flower and you crossed it with a white flower, you would get a pink flower. So that's a good illustration of what blending is. So let's just make sure we understand that they thought that the pink, or that which was blended, was something new and different, and that you couldn't get the originals back out. Just like, think about things that you blend. You guys blend a lot, okay? What do you guys blend?
Flour, water, bread.
You blend flour and water to make bread? What else do you blend? Like in your blender? Guys, I've never had these answers before. Peas and red peppers, yeah. So you're making hummus. I was thinking more of margaritas or something, yeah. It is spring break. That's exactly right. You are the culinary people, though, right? Okay. The point being, if I blend a smoothie, okay, which maybe is what? Yogurt and bananas and strawberries. If you blend that, can you ever get the bananas back out? No. So this is what they thought was happening, that when you blended these qualities, you got something new and different, okay? This man was Gregor Mendel. He lived from 1822 to 1884, and he did his work about that time, the mid-1800s. He is my idol. Okay, I love Mendel. Before we finish this discussion, hopefully you will have an appreciation for just how smart he really was. Okay, I believe he was probably one of the smartest humans that's ever walked the planet. Okay, he is considered to be the father of heredity. He gave us the fundamental laws of heredity, the basic principles of genetics, okay, that we still actually use today. He did his work in the 1860s, and again, he was an Austrian monk and math teacher, okay? It was his background in math that was so important. His abilities with regard to that field allowed him to make the discoveries that he did. Probably one of the first scientists to blend the fields of science and math, okay? But he did experiments with plant breeding. So he was crossing plants and observing how those traits were passed in the offspring, okay? So let's just talk a little bit about his work. he made some decisions up front that were very, very important, okay? He chose a plant to work with called the garden pea plant. Very important decision. Had he chosen any other plant, he might not have made the discoveries that he did, okay? So he chose this pea plant. Well, why did he choose it? Well, it was easy to grow, okay? Very important. And it had short generation times. Well, if you're doing crosses and you want to observe the offspring, it's really very nice to have a short generation time so you can have lots and lots of generations in a short period of time and do the analysis, okay? His pea plant also came in true breeding varieties, which was really important. So let's talk about what that really meant to Mendel. If a plant was true breeding at that time, it meant that if that plant was allowed to self-pollinate, that all of its offspring would have exactly the same characteristics that it did. Okay, so its offspring would be identical to itself if it was allowed to self-pollinate. Okay, in addition to all of these characteristics, another really, really important characteristic of this plant was the flower structure. Okay, keep in mind, he's crossing plants. He needs to control the pollination process. Okay, so let's take a look at this flower for a moment so you can get an appreciation for how difficult that might be. This flower was really prone to self-pollinate because of its structure. So let's get a basic background in some botany right now before we go on so we can understand the pollination process. Plants have sexual reproduction. So there is a female and a male, what we would say would be gametes. Okay? So this green portion in the middle of that flower is the female part of the plant, with the ovule, which would be the egg, in the very center of that structure at the base right there. The top of this structure, which is called a pistil, the top is called a stigma, and it's really, really sticky. Okay, that's where the pollen needs to land. The pollen is the male counterpart right here. The pollen is produced by this structure right there, that yellow structure called an anther. It's part of the stamen. So the stamen is the male, the pistil is the female. Okay, so what needs to happen is the pollen, if you were going to allow that flower to self-pollinate, the pollen from the anther needs to travel to the stigma, where the sperm cells would literally form a tube down to the egg or the ovule. Okay? So that's where the birds and the bees come in. Okay? You know, the bees are the pollinators, right? So if you wanted this flower to self-pollinate, all you would have to do would be cover the flower to make sure no other pollen got to it, right? But if I wanted to take this plant and cross it with another plant, I would have to ensure that the pollen from the other plant was the one that did the fertilizing. So what I would need to do would be snip off all those male parts, okay, and cover the stigma so that nothing would get to it, right? And then I would go get the pollen that I wanted from the plant that I wanted to do the cross with and make sure that that pollen was put right there on the stigma, okay? And then I would cover it up, right? So you got to control, to be able to control the cross, you got to control which pollen lands on the stigma, okay? A little bit about birds and bees there. Okay, the next thing he did was he kept it simple. Have you guys ever heard of the KISS principle? Keep it simple, stupid. Yeah, Mendel was the king of that. He kept it very simple. He studied very distinguishable traits. So he could just look at his pea plants and say, oh yeah, I know what it is. He didn't have to really study them. Here are the 7 traits that he studied. In his pea plants, this was very important, in his pea plants, he kept up with or analyzed these 7 characteristics. And they were very simple. I mean, how much simpler can you get than he had tall pea plants and short pea plants? There were no pea plants of medium height in this particular species. Okay? They were either tall or short. Only 2 options. In the flower color, there was only purple and white. As far as the pea color and pod colors, there was green and yellow. Okay? The peas and the pods came in two different textures. In the peas, you have some smooth peas, and then you have peas that are kind of bumpy looking, and he called them wrinkled. Okay? In the pods, they came in smooth, which he called inflated, or constricted. And as far as the stem orientation, there were plants that had terminal stem, which means that there's one main stem, or you have plants that do not have a main stem, and those are called axial. Okay? Those are the 7 traits that he studied. Very easy to just look at and know immediately what that plant is. Okay? Kept it simple. Probably the most important thing he did was his record keeping. Okay, keep in mind, he's a mathematician, so he's really into numbers. He kept very careful records. Take a look at a page from his journal. Can you guys see any of those numbers? You might can see just a few of them. Guys, I'm not talking about some monk who decided to cross a couple of pea plants one summer. This guy crossed hundreds and hundreds and hundreds of plants over years and years and years. Okay? And he kept very careful records of the differences in the physical traits of those plants and how many were produced. Okay? So he counted pea plants, hundreds and hundreds and hundreds of them. Okay? Without a computer. Imagine that. He kept up with all these records. Okay, so his first crosses were called monohybrid crosses. Monohybrid crosses. And we call it monohybrid inheritance. What does mono mean? One, and the word hybrid literally means a cross. Okay? So what he was doing was he was taking plants that were two plants that were exactly identical in every respect except one. There would be one difference between the two plants. So he's cross-pollinating plants that differ in one trait. He's cross-pollinating plants that differ in one trait. And then he would observe that single trait and how it was passed to the offspring. And then he would cross two of the offspring and observe how that trait was passed to the next generation of offspring. Okay? And to make matters worse, he had to do everything double. Okay? He had to make sure every cross that he did was reciprocal. Okay, now what in the world does that mean? Well, as I mentioned earlier, he had no idea what the female contribution was versus the male contribution. He had no idea. He didn't know, was the female more important than the male? Was the male contribution more significant than the female? So everything he did, he had to do it reciprocally. Okay, example. Let's say that both of these plants that you see there are exactly the same in every respect except for one trait. This plant over here to your left is going to be a tall plant, and this plant is going to be a right plant on the right. I mean, a short plant on the right, okay? So for every cross he did, he had to do it back and forth. For instance, he would take the pollen from the tall plant and use it to fertilize the ovule of the short plant. And then he had to do it the other way. He had to take the pollen from the short plant and fertilize the ovule of the tall plant because he didn't know if it would make a difference or not. Okay? So his very, very first crosses were always true breeding plants. And he called those first crosses his parents. That's his pea generation. So the very first cross was always true breeding. Those were his parents. Now, what was a true breeding plant? If it was allowed to self-pollinate, the offspring would be exactly like it. Yes, so that's a true breeding plant, okay? So he would cross those two plants, and the offspring, the result of that cross, he called his F1 generation, or first filial generation. Filial means offspring or family. Okay, so he takes his P generation, he crosses them. The next generation is his F1, right? That's his first generation of offspring. Then he would cross two of his F1s, and the result of that would be his second filial generation, or his second generation of offspring. Everybody got it? Parents are true breeding. The result of that cross is his F1. He crosses two F1s, and the result of that cross is his F2. Okay? Okay, guys, he did this with all of those traits. He would do this with height. He would do this with flower color, pea color, pod color, etc. He did it with all of those traits numerous times. Okay? And he started noticing a trend. He noticed that for every trait that he did this with, the F1 appeared to be all one of those characteristics. For instance, when he crossed purple flowered plant with a white flowered plant, all of his F1s had purple flowers. The white disappeared in the F1 generation. When he crossed tall and short, all of his F1 were tall. Shortness disappeared in the F1 generation. So he did that with all of those traits, and for every one of them, one of the characteristics disappeared. All of the F1s were the same. Okay? He also noted that when he crossed two of his F1s, that which had disappeared always came back in a 3-to-1 ratio. So if we're talking flower color, the white disappeared in the F1, but came back in 25% of the F2. 3-to-1 ratio. Okay? Tall and short, short disappeared in the F1, but in the F2, 25% of the offspring would have been short. So that which had disappeared always came back in a 3-to-1 ratio. Okay? So after analyzing this, he came up with a theory of what he thought was happening. Now, guys, I want you to always remember, this is 1850 and he's a monk, okay? Okay? And he's counting pea plants, okay? He came up with a theory about what he thought was going on, and he called it the particulate theory of inheritance. Now, what the heck is a particulate? Have you ever heard that word before? Particulate? You culinary people, y'all probably blend them, don't you? I love y'all. Okay, a particulate sounds something very close to a particle, right? Except maybe smaller. Particulate, something very, very small. He had no idea what he was dealing with. He just said there's some kind of little particle, okay? And he actually called those little particles factors, okay? And he said, now keep in mind, he counted pea plants, okay? He said, I think that for a pea plant, every trait in that pea plant, okay? So each trait, what's a trait? A trait is height, flower color, pea color, okay? So every plant for every trait has two of something that's causing it to have whatever it has, okay? So each trait is determined by a pair of factors. So if a pea plant's tall, there's two of something that pea plant has that's causing it to be tall. If a pea plant is short, there's two of something that's causing it to be short. And more importantly, guys, he said, whatever these are, they're passed from parent to offspring unchanged. Think about that for a minute. 1850, he just said something's going from parent to offspring, and it's not changing. There's no blending happening. Okay? So he just blew the blending theory of inheritance out of the water. He said, no, nothing's blending. It's whatever it is, it's being passed from parent to offspring. Okay? So let's think about that for a moment, okay? Let's take Mendel's example of pod color. Okay, remember we said pod color is either yellow or green, right? Well, Mendel said there must be factors causing greenness. Okay, so when a plant has green pods, it has two of those factors. Okay, so let's use big G to stand for green. Okay, so he said if an individual has two green factors, it's going to have green pods. Okay, and then there has to be a yellow factor causing those individuals with yellow pods to be yellow, right? So let's just use little g for yellow. Okay, so if a plant has yellow pods, it's got two yellow factors. If a plant has green pods, it's got two green factors. Okay, but he said, wait just a minute. What if a plant has one of each? Okay, what if it has a green factor and a yellow factor? What color would its pods be? Okay, so the more he thought about that, he said, okay. He said, I can explain my F1 results. What was F1? What happened? They were all the same. He said, these factors, one of them's going to hide the other one. He said, I'm going to say it's dominant. So he came up with his theory of dominance, okay? And he said, of course, if a plant has two green factors, it's got green pods. If it's got two yellow factors, it's got yellow pods. But when it has a green and a yellow factor, one of those is going to dominate, okay? So when an individual has two factors that are not alike, one dominates the other one. The other one is still there, but it's just not showing, okay? And he called it his theory of dominance. For instance, if you've got a plant that has two green factors here and a plant that has two yellow factors and you cross them, then the offspring are going to get one of each, okay? And they're going to look like that, right? So he said the offspring have a green and a yellow factor. The offspring appear to be green because green is dominating yellow. The yellow factor is still there. It's just being covered up by the green factor. Okay? Today we call the yellow what? Recessive. That's exactly right. Okay? Mendel counted pea plants. Okay? Okay. So then he said, wait, okay, this is making sense. Now I can explain my F2. What happened in F2? Huh? The one that disappeared came back in a ratio of 3 to 1, right? He said, look at this. These factors segregate during gamete production. So I'm going to call it my theory of segregation. Okay, my law of segregation. He said, when an individual produces gametes, now, what's a gamete? Egg and sperm, right? When an individual produces gametes, the factors separate so that every gamete gets one. Okay, look at this. So if this is which generation would this be of Mendel? This is going to be the F1, right? I'm crossing two F1s, right? So I'm coming up with my F2 generation, okay? So when this individual is producing pollen and eggs, these two factors are going to separate so that every gamete just gets one of them. So this individual is going to produce some gametes that get the big G and some gametes that get the little g, okay? So each gamete is going to get one of the factors. When this individual produces gametes, some of the gametes are going to get the big G, and some of the gametes are going to get the little g, right? Okay? So Mendel said, when I'm crossing these two plants, anything could happen. When you talk about fertilization, there could be some times when this gamete that has the big G fertilizes this gamete over here that also has the big G, right? So then what would my offspring be? Big G, big G. So it would be that one right there, wouldn't it? Right? So some of the time, this gamete of the first individual is going to fertilize the gamete that has the little g in it of the second individual. So what would my offspring be? Big G, little g. And then some of the time, the first individual's gamete that has the little g is going to fertilize the second individual's gamete that has the big G. So what's my offspring? And then some of the time, it will happen that the first individual's gamete that has the little g is going to fertilize the second individual's gamete that has the little g, and what's the offspring? And look at your ratio. That one's green, that one's green, that one's green, and 1 out of 4 are yellow. Okay? So he explained his F1 generation by understanding what dominance was, and he explained his F2 by recognizing that these factors separate in gamete production, right? Okay, so let's summarize, okay? Mendel said each individual has 2 factors for every trait. Mendel said factors segregate during gamete formation. Mendel said each gamete contains 1 factor from each pair. Mendel said fertilization gives you a new individual, the offspring, that has 2 factors for each trait. Guys, look at that. That's 1860. This is a monk who counted pea plants. What did he just explain to us? Do what? Yeah. Meiosis. Yeah. Okay? So now we know, as John said, now we know that Mendel's factors were really the alleles of a gene. Okay? Remember, genes are on chromosomes, right? And you have 2 of a kind. The gene has alleles. You have homologous chromosomes, right? You would have 2 alleles for a gene, and one of those you would get from your mom and 1 from your dad, right? So he's really talking about the genes of which the alleles are on homologous chromosomes. Okay? Mendel's talking about genes. And so what do we know happens to homologous? What happens to those alleles for that one gene? What happens to those alleles in meiosis? They separate. That's exactly right. Mendel says factors, alleles, separate during gamete production. He just described meiosis 1. I mean, we've said for weeks now, what's the purpose of meiosis 1? The homologous pair separates, right? Mendel said each gamete contains 1 factor. That's the definition of haploid. And then he says when fertilization occurs, the sperm fertilizes the egg, you restore that condition where the new offspring has 2 factors. That's diploid. Can you believe he figured this out? I mean, think about it for a minute. You've got a textbook with lots of explanation. We have movies we watch to help us understand meiosis. We played with funny little beads and had fun doing it. And sometimes we still have a problem understanding meiosis and what happens. And this man figured it out counting pea plants. Okay, so let's get some vocabulary down. So we can move on. What you're looking at there is a pair of homologous chromosomes. Remember, let's go back and review for a moment exactly what a chromosome is. Remember, we said a chromosome has genes on it. We said this would have been chromatin, single-stranded, right? And during interphase, chromatin actually replicates, makes a copy of itself, of which the copy is held together by a centromere, right? Okay, so whatever is on this chromatin, sister chromatid, is exactly identical to what's on the other sister chromatid, right? So they're identical to each other. And these are homologous chromosomes, so they have the same genes for the same traits and the same locations. So if this is the gene for height, okay, those are alleles for short, and those are alleles for tall, right? You guys remember all that from last time we were here like 2 years ago? Okay? Okay. So an allele is the alternate form of a gene. It's the form a gene takes. Mendel was very fortunate that his traits were controlled by 2 alleles. That's not always the case, and we're going to talk about that later on, but he was very fortunate that height had 2 alleles. 1 gene, 2 alleles controlling it. One from the father, one from the mother. So now we can think about a chromosome, that structure, as just a linear arrangement of genes. Okay, the genes are on the chromosome, and where they are located is actually called a locus. The gene locus is the location of a gene. I think we've discussed a little bit earlier that not every base pair of your DNA is involved in a gene. So you're going to have quite a lot of this chromatin or chromosome that's actually not included in a gene. So your genes have very specific locations, and wherever the genes are on one chromosome will be exactly the same place that that gene would be located on its homologue. Okay, so there's your gene for height. It's in the same location on that chromosome. Okay, Mendel's word dominant is one that we still use today. As you mentioned a while ago, the opposite of that would be recessive, right? Okay, so dominant, the word dominant, we use to describe the trait that is expressed. So dominant is the trait that's expressed. When you have an individual that has unlike alleles or factors, as Mendel would call them, that which is expressed is referred to as the dominant one. Okay? It masks or covers up another allele. And when we're representing a dominant allele, we always use a capital letter. So for flower color, we would use a big P to indicate purple, okay? So if I were to show the alleles for that flower at the top, it would be big P, okay? White is recessive, or the allele that's covered up, okay? The allele that's actually masked when a dominant allele is present. And we would use the lowercase of that same letter to show that it's recessive to purple, okay? So to illustrate or signify the white allele, we would use a little p. So big P would be dominant, purple. Little p would be white. Okay? Everybody good? If an individual has 2 alleles that are alike, okay, we would call that individual a homozygote. Homozygous is when an individual has 2 identical alleles for a trait. Okay? So that first individual there at the top of the illustration, this would be considered to be a homozygous dominant individual. When I say homozygous dominant, homo means the same. That means that individual has 2 of the same alleles for that trait. And they're the dominant alleles. Okay, if I say homozygous dominant, that means 2 alleles that are exactly alike, and they are the dominant alleles. So that would be the big P, okay? This individual to your right, little p, would be called a homozygous recessive individual. Homozygous meaning that the alleles are exactly the same. Recessive telling me that it's the little p. So homozygous recessive would be 2 little p's. Okay? An organism that is homozygous for a trait will only express that trait, that characteristic for that trait. If it's a recessive allele, it's going to show the recessive appearance. That's all it's got. Okay? If it's the dominant allele, it's going to show the dominant appearance. Okay? If an individual has 2 alleles that are not alike, we call it a heterozygote, or it would be referred to as being heterozygous. Okay? Hetero means other, just like homosexual and heterosexual. Okay? Hetero means other or different. So individuals that have 2 unlike alleles would be heterozygous. And keep in mind, if an individual is heterozygous, that means that one of them is going to show in its appearance and the other one is not. It's hidden. It's recessive. Okay? So one trait will express and one won't in a heterozygote. We good? Okay. The alleles, the appearance of an individual is called its phenotype. Think of pheno as physical appearance. Okay? So the phenotype of an individual is what appears. Okay? So what are some examples of phenotypes? Phenotypes would be purple and white. Phenotypes would be tall and short. Phenotypes would be green and yellow. Phenotypes would be blue eyes and brown eyes. Okay? The genotype, think of gene. The genotype would be the alleles that are producing that phenotype. Okay? So the genotype are the alleles that individual has that causes that phenotype. Okay? So genotypes would be 2 dominant alleles, a dominant and recessive allele, or 2 recessive alleles, right? So it's either homozygous dominant, heterozygote, or homozygous recessive. Those are genotypes, okay, that produce physical appearance. Okay? Something that you need to make note of, this is really important that you understand this, is that if an individual is showing the dominant phenotype, what does that mean? If the appearance of the individual is the dominant, okay, in this example, purple, that phenotype could be either homozygous dominant or a heterozygote. You will not know that just by looking at it. Okay? So you can't look at a purple flower and know if it has 2 dominant alleles or a dominant and a recessive allele. So dominant phenotypes could have either or. Okay? But if an individual is showing the recessive phenotype, in this case, whiteness, that recessive phenotype is only always the result of 2 recessive alleles. There's only one genotype, homozygous recessive, that can result in the recessive phenotype. Very important that you understand that. Got it? Okay. 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 Oftentimes, we want to predict what the outcome of a genetic cross might be. And to do that, we would use something called a Punnett square. How many people have ever done a Punnett square? Very good. That's like 3/4 of the class. Good deal. Okay. Punnett squares, actually, I've been using them the whole time we've been talking. All of my examples have been Punnett squares to kind of get you used to looking at them. It's just a chart that will show you all the possible genotypes and phenotypes of a genetic cross. Okay? And guys, I know you're sitting there probably saying, what's the big deal? Why would I want to know what the cross between a purple flower and a white flower would be? Okay, well, that's probably not that important. But if you have $1 million racehorse and you're getting ready to breed it, you definitely want to know what your outcomes could be before you take the risk of putting that horse in jeopardy of breeding it and producing an offspring. Okay, so we would want to do this chart that's going to show us all the possible offspring. Guys, this is all about probability. Not what will happen, but what could happen. Okay? So, what do? Well, we are going to take all the possible sperm genotypes on one side and all the possible egg genotypes on the other side of the square, and then we're going to cross them and get all the possible zygote genotypes. Okay? So we're going to take all the possible sperm that could be produced and all the possible eggs that could be produced, and we're going to act like they're fertilizing each other to see what all the possible offspring could be. Okay? So let's take a look at this example. Okay? If that's the genotype, big B, little b, of one of my parents, and that's the genotype, big B, little b, of my second parents, I'm going to act like they're producing gametes. Okay? So I'm going to say, if that individual produced gametes, remember what happens to those alleles in gamete production? They separate into gametes, right? So some of my gametes are going to have the big B in them, and some of my gametes are going to have the little b. Okay? So I'm going to put those at the top, and it doesn't matter which one you put where, you're going to get the same results. Then my second parent also has the same genotype. So when this second parent produces gametes, those alleles are going to separate. So I'm going to put the big B gamete right here on the left and the little b gamete right there. So basically what I've done is this is one parent at the top, the gametes that could be produced. This is the other parent on the side, the gametes that could be produced, and I'm going to say if this big B gamete fertilizes that big B gamete, my offspring would be that. If my big B gamete from my female fertilizes my little b gamete from my male, my offspring would be that. If my little b gamete from my female fertilizes my big B gamete from my male, that would be my offspring. And if my little b gamete from my female fertilizes my little b gamete from my male, my offspring would be little b, little b. So I've been using that this whole lecture. You just didn't know what it was yet. Okay? That's a Punnett square. So let's just go through some very basic steps. For those of you that have never done this before, this is the logic. We're going to get in your notes everything that I just said now, okay? This is the logic that you would follow in doing a Punnett square. Now, some of you that know how to do this is going to seem very simple to you. But don't forget the basics, because when we advance to the more complicated Punnett squares, you might consider the basics very helpful, okay? The first thing is you're going to be given a problem. You're going to be given a problem. There's the problem up there. Cross a heterozygous tall plant with a heterozygous tall plant. Okay? It's basically the same problem we just did on the other screen. To be able to work a Punnett square, you have to know which characteristic is dominant and recessive, and I'm always going to tell you that. And as a matter of fact, I'm going to give you the symbols to use because I want everybody using the same symbols. It makes it easier to grade, okay? So I'll tell you up front, for instance, use big T for tall and little t for S, okay? When I tell you that, that automatically tells you tall's dominant, right? And short is recessive, okay? So you're going to take those symbols and you've got to understand what the words are to be able to work the problem. The next thing you're going to have to do is figure out the genotypes of the parents. So what does it mean, what is the genotype of a heterozygous tall plant? Huh? What would the alleles be? Big T, little t, right? That's a heterozygous tall plant. Well, both of my parents have the same genotype. Guys, something that was always very helpful to me was my teacher told me the width separates the parents. Okay? So if the words start confusing you, and when we get into the more complicated problems, they may, if the words start confusing you, always find the width. The width is your cross. Okay? So a heterozygous tall plant crossed with a heterozygous tall plant. So that's your problem. Make sure you always write your problem. Okay? If you mess up from there on out, I can give you partial credit for that because you understood the words. Okay? Okay? Okay, so the next thing you have to do is determine the gametes. Well, that individual can produce gametes that have those in them. Big T, little t will have some gametes with the big T. It will also have some gametes with a little t. The other parent is big T, little t. That parent will have some gametes with the big T and some gametes with the little t, right? Then all you have to do with the gametes is set up your Punnett square. So you're going to take these gametes and put them at the side, and take those parents' gametes and put them at the top, and then do your cross. Okay? So then you're done, except for just deciding what you got. Okay? Sometimes I may ask you about the genotypes. So if I wanted to look at the genotypes and the ratio, I would just count them. I would say that's a homozygous dominant, that's a heterozygote, that's a heterozygote, and that's a homozygous recessive, right? So I have one homozygous dominant, two heterozygotes, and one homozygous recessive. So I've got a 1 to 2 to 1 ratio. What about the phenotypes? Big T would be what appearance? Tall. Big T, little t would be what appearance? Tall. Big T, little t would be what appearance? So I got 3 talls, and little t is short. So I got a 3 to 1 ratio. Yes. Okay? Remember a while ago I said you can't tell what the genotype of an individual is who has the dominant phenotype? Okay? We were talking about purple flowers, and I said a purple flower could either be homozygous dominant or a heterozygote, right? So let's take a look at this right here for just a minute. If I've got a tall plant, what could its possible genotypes be? It could be big T or big T, little t. Remember, we can't tell by just looking at it, right? But the short plant, what's its genotype? Little t, okay? So a way that, something I could do to find out what the genotype of the tall plant is if I cross the tall plant with the short plant that I know for a fact has to have little t, I can find out what the genotype of the tall plant is, okay? This is called a test cross. This is a monohybrid test cross. And you do test cross to find out what the genotype is of an individual that exhibits the dominant phenotype. If you have any, yes, sir. 4. Oh, let me show you the results. Okay, let me show you the results. Okay, Austin, I want you to think about this for a minute. Okay, if my tall plant, it could be either one of these, right? Can't tell by looking at it. If my tall plant is a homozygous dominant, think about it for a minute, okay? All it has are dominant alleles, right? So when it produces gametes, every gamete is going to get a dominant allele for height from this tall plant, right? So that means that the offspring of this plant, if it's homozygous dominant, will always be tall. Does that make sense? If all this tall plant has is dominant alleles, that means all of its offspring will be tall, always, no matter who you cross it with, because all it has is dominant. Okay? But think about this. If my tall plant is a heterozygote, that means that some of its gametes are going to contain the little t, right? Some of them. So that means that some of the time there is a possibility that the gametes from this tall plant that have the little t in them are going to fertilize the gametes from that short plant. So some of my offspring are going to be short. So basically what we're saying is if you have this individual that has the dominant phenotype, if you cross it with an individual that has the recessive phenotype, if any of the offspring show that recessive phenotype, this parent, the tall parent, has to be hiding a recessive allele. Does that make sense? That's the only way it can happen. If you have a tall plant that produces a short offspring, it has to have a short allele. Okay. Any questions? There's your Punnett squares. There's your Punnett square if the tall plant's heterozygote. And there's your Punnett square if it's a homozygote dominant.